Good evening, everyone. I hope I am clearly audible and my screen is visible to you. So, today we will be discussing again with week 5 contents on the course of Introduction to Cell Biology, Cell Components, Organization, and Processes. So, this is a NPTL course which is being floated from January to March. Mm. So the course instructor is Professor Shikha and I am the one of the teaching assistants. I'm Ananya Chatterjee. I'm a PhD student in ISC Bangalore and also a Prime Minister Research Fellow. So today's class we'll be basically discussing about the week 5 contents, which is mostly meiosis and nuclear localization. So will be starting with so we we will be starting with the assignments which was given to you in the previous week. The first question is the differential localization of ran GTP and ran GDP in cell compartments is important because it helps in the formation of nuclear pore complex. It provides directionality to nuclear import. It helps in recognition of the NLS, none of the above. So, as you have already went through those, as you have already gone through those lectures in the portal, but also today we'll be discussing more about this nuclear localization, how RAN GTP and RAN GTP helps. So, for answering this question, if you know that. So whenever any protein or any macromolecules wants to go to nucleus, so there is a mechanism going on which is also called nuclear import and nuclear export. So in this nuclear import and nuclear export process, what happens? RAN GTP and RAN GTP plays an important role in phosphorylating and dephosphorylating. So this RAN GTP and RAN GTP, there is a differential localization present in the cell compartment. So uh, So this differential localization, how it helps, it basically gives a directionality to nuclear import. So how it helps, we'll discuss in the next slide. But you have to know that it gives a directionality. It doesn't help in the formation of nuclear pore complex, nor it helps in recognition of NLS. What is NLS? Nuclear localization signal. So this is a different aspects. We'll cover it. The next question is, which of the following options correctly describe an event occurring in prophase 1 of meiosis? So, what are the options? Pairing of homologous chromosome and crossing over, alignment of homologous chromosome at the cell equator, duplication of DNA, segregation of homologous chromosomes. So when we'll go through in details what are the stages of prophase 1 of meiosis, we'll see that homologous chromosomes come closer with each other, okay, and they pair to each other, and then they eventually do crossover. So this the whole process happens in the prophase 1. So option 1 will be the correct answer. The next question is, pick the correct order of different stages in meiotic or meiotic prophase. Options are leptotene, zygote, pacatine, diplotin, diakinesis. So you have to remember that the stages are subsequently leptotene, zygote, pacatine, diplotin, and diakinesis. We'll discuss about what are the process happen in these stages. So as I was telling, nuclear protein import at the help of RAN GDP and RAN GTP. So here you can see this is a nucleus, this is cytoplasm. So This is a protein. TRPS1 is a transcription regulator or repressor. This protein is a nuclear protein, so it needs to get imported to the nucleus. So it should have one nuclear localization signal in this protein. Along with that, to get it import in alpha or import in beta, something like that will help and mount with the this protein and will go. But for that, you have to have RAN GDP complex or RAN GDP complex, uh, a differential equilibration you should have in the cytoplasm and nucleus, so that it can phosphorylate and or dephosphorylate accordingly. Okay. So when the protein is getting imported into the nucleus, what is happening? You can see here RAN GTP is present. So RAN GTP is uh, getting bound with the importin also. So it's getting so the importin alpha and TRPS gets released, and RAN GTP is getting bound with the importin beta, and then it importin beta is getting exported to the outside of the nucleus, that is cytoplasm. And also that thing, when this RAN GDP is forming RAN GDP again when one phosphate gets out, and also when this dephosphorylation happens, this important alpha also get released to again bound with the TRPS1. So this is how the equilibration happens, and this is how the import export happens through nucleus. As we were discussing about different stages of meiosis, so we know that in meiosis there are some stages like mitosis, prophase, metaphase, anaphase, telophase. But in prophase is a very long phase in meiosis, and this prophase is consisted of another 5 small phases, which are leptotin, zygotin, pachytin, diplotin, and diakinesis. So what happens, we'll discuss in a brief in these stages. Leptotin, what happened, the nuclear membrane will start getting degraded, and after replication in S phase, now what will happen, chromatin will now start condense, and this will form chromosome. So here you can see this two chromosomes are visible here. So one is pink purple color, one is green color. So those are homologous chromosome, you can say, and in one chromosome you can see two chromatids, those are sister chromatids. But in two homologous pair, you can those two chromatids are called non-sister chromatid. Okay, so this will be important in understanding the later stage. So in zygotin, what happens, those homologous pairing will start. So that is, these two chromosomes are homologous to each other, so they will come closer, such that around 100 nanometer closer they will come, and they will start pairing as homolog, and they will, so to get it paired, these two chromosomes, there should be some protein complexes which will hold these chromosomes together. So those complexes are called synaptomial complex, and this synaptomial complex formation starts from zygotin itself. Then the pachytin stage, what happens in this stage, SC formation is successfully happened, synaptomial complex formation, and also crossing over starts. So crossing overs means the exchange of chromosome or exchange of chromatids between two homologous chromosomes. In diplotin, crossing over completes and SC disassemble. So this, when this crossing over is completed, then this synaptomial complex should be disrupted. So this disassembled Homologs stay connected only at chiasma. So in this stage, what happens? The homologs will stay connected on the chiasma. What is chiasma? Chiasma is the place where the crossing over or the recombination happened. So here only one side is visible as a chiasma, but in a chromosome multiple sides can be crossed over, and also these steps are very regulated process. In diakinesis, what happens? Homologous gets separated and also nuclear membrane dissociates. So now we will come back to next question. Synaptomial complex disassembly takes place in. We have gone through just now that the complex disassembly takes place in diplotin stage. Non-disjunction due to improper segregation of chromosomes may lead to what? Aneuploidy, Down syndrome, progeria syndrome, none of the above. So non-disjunction, what is that? Now we are seeing that this homologous pair of chromosome, it will go like that to the next step. So what happens from meiosis 1 should be at the another pole. So if there is something happens that in meiosis 1 only this sister chromatid gets segregated, or only both the chromosomes are going into another egg, or another gamete. So what will happen? This will lead to aneuploidy or polyploidy, something like that. So the answer will be as well as aneuploidy as well as Down syndrome. So because Down syndrome also we see this kind of misarrangement of chromosomes, or this kind of improper segregation. So the answer will be both. The next question is, during homologous recombination, the point at which the chromatids from a pair of homologous chromosomes are physically attached to each other is called, we have just gone through chiasma. So the point there will be the point where the homologous pair of chromosomes are getting attached is called chiasma. In budding yeast, the next question, in budding yeast, the meiotic commitment initiator IME acts as IME, also called inducer of meiosis. So IME also known as inducer of meiosis. So this gene, which is required for sporulation and sporulation specific gene expression. So this is basically what helps in gene replication or gene expression. Those are called transcription factors. So this IME is a transcription factor. Now the next question is, the proteins that are targeted to nucleus have a signal sequence that has sequences rich in positively charged amino acids, sequences rich in negatively charged amino acids, sequences rich in uncharged amino acids, sequences rich in aromatic amino acids. So nuclear localization signal or sequence is a amino acid. So whenever a protein needs to get imported inside the nucleus, so we have talked that there should be some signal which will help those protein to go to their destination or designated places. So in the case of nucleus, there should be some stretch of amino acids which are basically lysine and arginine rich sequence. So here which will go with the logic is that positively charged amino acids. So as lysine and arginine are positively charged, so answer will be A. The next question is, pick the correct statement regarding nuclear organization. The heterochromatin region is darkly stained and transcriptionally active. Euchromatin is lightly stained and transcriptionally active. Histone Histone deacetylation by SAR2 causes euchromatin formation, all of the above. So the correct answer is that this is not the correct answer. Heterochromatin region is darkly stained, this is fine, but it is not transcriptionally active, rather the opposite one. The correct answer will be euchromatin is lightly stained and transcriptionally active. Okay. Now we'll talk about a little bit more of nuclear localization signal. So here you can see this is a T antigen which needs to be imported inside nucleus. So it has a normal nuclear import signal, that is lysine and arginine rich signal. So and it is localizing inside the nucleus. But when someone has mutated this sequence, what happens that the localization of this T antigen is getting hampered, and these are localizing in cytoplasm itself. So this is a function proved, the function of nuclear localization proved by this experiments, where you can see these are immunofluorescence micrograph. So basically shows the cell location of SV40 virus T antigen containing and also lacking a short sequence that serves as a nuclear localization signal. The next question is that the cytoskeleton supporting the nuclear membrane is made up of tubulin filament, myosin filament, lamin filament, none of the above. So nuclear membrane, so there is a cytoskeleton which supports just underneath the nuclear membrane is called lamin filament. So yes, so the answer will be lamin filament. So in this picture you can see this red colored proteins are lamins, so which are basically supporting the nuclear membrane. So we'll briefly start about meiosis and we'll go through slowly. So what is meiosis? Now so how it differs from mitosis? Mitosis basically helps in dividing cells, but meiosis or meiosis will help to increase the progeny. So what happens, this is a form of cell division by which gametes with half the number of chromosomes are produced. So from diploid cell, one haploid gamete, from diploid cells two haploid gametes are produced. So it's also called reductional division. Meiosis is a sexual reproduction. Two division, meiosis is consist of two division, meiosis 1 and meiosis 2. So it maintains a consistency in the chromosome number from generation after generation. In the absence of meiotic cell division, the chromosome number of a species would be doubled in every generation due to the fusion of male and female gametes, an impossible biological situation. So meiotic cell division is a very much important process in our body. So this is pre-meiotic interphase. What happens? Chromosomes replicate in the S phase. So this is a single stranded chromosome which undergoes a replication in S phase and forms double stranded chromosome. Each duplicated chromosome consists of two identical sister chromatids attached to the centromeres. Centriole pairs also replicate. Nucleus and nucleolus visible. Okay. Now we'll come to first meiotic division or meiosis 1. As I was discussing that it also consists of 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1. So in meiosis 1 what happens? Cell division occurs that reduces the chromosome number by 1/2. Prophase 1 is the longest and most complex phase of meiosis 1. And it also consists of 5 substages. These are leptotin, zygotin, pachytin, diplotin and diakinesis. In leptotin what happens? There is a marked increase in the nuclear volume. So, and also there is chromosome condensation so that they become visible as fine threads like a ball of knitting wool, each chromosome of two chromatids. Okay, so now the nuclear volume becomes increased as the nuclear envelope will get, or the nuclear membrane will get disrupted, and also now chromosome condensation starts forming. So each chromosome consists of two chromatids after condensing. Now zygote is the second stage of Tropus 1. A diploid cell contains two sets of chromosomes. The two chromosomes similar in size, form, structure are called homologous chromosomes. One of the homologous chromosome is parental and other one is maternal. During zygote, these two homologous chromosome starts pairing each other, and these homologous chromosomes come to lie side by side in pairs, and this pairing is also called synapsis. And the complex formed by a pair of synapse chromosome is called bivalent. Electron microscope studies of this stage shows that synapsis is accompanied by a structure called synaptomial complex, which is thought to stabilize two homologous chromosomes till the crossing over is completed. This is homologous chromosome. This eye color locus. So uh, what is stating is that these two chromosomes are same as function and the location of the genes, but they are coming from one from maternal and one from paternal. Now what is happening in the packeting stage? In this stage, the synapsed chromosome becomes thick and short, and the chromatids of the homologous chromosome now become clearly visible as tetrads. Tetrads how? One homologous chromosome consists of two chromatids. So two homologous chromosomes lay side by side and they are forming four chromatids. So that is tetrad. During packeting, crossing over takes place between non-sister chromatid of homologous chromosomes. The exchange of genetic material between non-sister chromatid of homologous chromosome is also called crossing over. So we'll know this in great details in future slides. Crossing over leads to recombination. So crossing over, when this homologous chromatids gets crossover, what happens? It leads to recombination of genetic material, which involve a mutual exchange of the corresponding segments of non-sister chromatids of homologous chromosome. And this recombination or the crossing over happens by breakage of the bond of DNA or the chromosomes, and reunion of chromatid segments occur. The site where crossing over occurs forms a replication nodule. Recombination nodule. So this recombination is an enzyme mediated process. An enzyme also called recombinase helps in this process. So by the end of packeting, the recombination between the homologous chromosome is now complete, and the two chromatids are linked at the site of crossing over. Okay. So this is how the process is going. Now in diploid, what happens? When we are done with the crossing over, what will happen? The two homologous chromosomes begin to separate it, and the synaptomial complex which was holding together now will start dissolving. So the point of attachment between the homologous chromosome after dissolution of the synaptomial complex is called chiasmata. So from all the sides the synaptomial complex will get disassembled, but where only the crossing over happened, that side will be attached with each other, that is called chiasmata, and it becomes visible during diploid stage. So the two homologous chromosomes begin to separate from each other but remain attached at the chiasmata at diploid stage. So chiasmata mark the site where the crossing over has been occurred during packeting. The homologous chromosomes do not separate at chiasmata, hence they form a X-shaped structure. Now diakinesis, it represents transition to metaphase 1. The following events occur. Spindle assembles to prepare homologous chromosome for separation. It assembles at the poles so that it separates the homologous chromosomes into two cells. Terminalization of chiasmata. Due to tight condensation of chromosomes, the chiasmata disappear from the chromosome by slipping off or sliding from the tip of the chromosome, and from there it either slips off or remain at the tip. Nucleus Nucleolus disappear and the nuclear envelope also starts disintegrates. Now metaphase 1 in brief. Bivalents are arranged at the metaphase plates. Centromeres of the two homologues of each bivalent on the either side of the equatorial plane. Metaphase terminates as soon as homologous chromosomes begin to separate from each other. Okay, and they will migrate to the opposite fold. So these are homologous pair of chromosome, you can see. So now this homologous pair of chromosome will get separated from each other. In anaphase 1 they will start separating, you can see. So this separation of the homologous chromosome happened, and then one chromosome from each bivalent begins to migrate to the other pole. As a result, the number of chromosome each poles becomes half. So this will become from 2n to n. In telophase 1, the chromosomes uncoil only partially. The nuclear envelope again starts making and nucleolus also starts reappear. Now there will be cytokinesis, where there will be formation of the cell membrane, which will form two gametes or two cells. Now the next step is interkinesis or intrameiotic interphase. Interkinesis is a metabolic phase between telophase 1 and prophase 2. During this phase, chromosomes are elongated but do not form chromatin fibers. There is no replication of DNA during this phase, but centrioles pair replicate in an animal cell. The RNA and protein required during meiosis 2 are also synthesized during this phase. Now there will be meiosis 2. During meiosis 2, two sister chromatids of each chromosome separate and migrate to the opposite pole. So meiosis 2 resembles with the mitosis. So then what will happen? Each chromosome will two sister chromatids from each chromosome will get separated and will go to opposite pole. As a result, the number of chromosomes in each of the two haploid nuclei remain the same, that is haploid, at the end of this division. The second division of meiosis is often referred to as equatorial or the equational division, because it is forming n to n only. So this is also called meiotic mitosis. The second meiotic division is also divided into four stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. And the four stages of meiosis 2 is same as mitosis. So if we compare meiosis with mitosis, so what happens? Meiosis is a form of nuclear division in which a single round of chromosome duplication is followed by two rounds of chromosome segregation. The duplicated homologues, each consisting of tightly bound sister chromatids, pair up and are segregated into different daughter nuclei in meiosis 1, and the sister chromatids are getting segregated in meiosis 2. What will happen in meiosis 1? There will be genetic recombination, also called crossing over. So each diploid cell that enters into meiosis, therefore producing four genetically different haploid nuclei, which are distributed by cytokinesis into haploid cells that differentiate into gametes. In mitosis, by contrast, homologues do not pair up, and the sister chromatids are segregated during the single division. Thus, each diploid cell that divides by mitosis produces two genetically identical diploid daughter nuclei, which are distributed by cytokinesis into a pair of daughter cells. So in the mitosis, the difference is that there is no crossing over, there is no genetic recombination, there is nothing of homologous pairing, but what will happen? Now each diploid cell will divide and will make two other diploid cells. and that will be also most important, that will be genetically identical because there is no crossing over happening. So in meiosis, you can see this is a parental homologue, this is a maternal homologue, that is paternal homologue, maternal homologue comes from further comes from mother. DNA replication is happening, so one chromosome is forming two chromatids. Pairing and crossing over of duplicated homologues will happen. So homologue will where you can see this gene location are same, those are basically homologue pairs. They will come to become and line up on the spindle. Then segregation of the homologues will happen at anaphase 1, and before that there is, you can see at the chiasma the crossing over has been started and will go on. In anaphase these two homologues will get separated, and after telophase this is the scenario. Then in the segregation of the two sister chromatids, hmm. So what happens in meiosis 2? Segregation of two sister chromatids at anaphase 2 will occur. So these two sister chromatids also get segregated and they will make four haploid daughter cells. So duplicated homologues pair during meiotic prophase. So what happens during mitosis in most organisms, homologous chromosomes behave independently of each other. But during meiosis 1, it is crucial that homologues recognize each other and associate physically in order for the maternal and paternal homologues to be bioriented on the first meiotic spindle. Spatial mechanisms mediate this interaction. The gradual juxtaposition of homologues occur during a prolonged period called meiotic prophase, which can take hours in yeast, days in mice, and weeks in higher plants. Like their mitotic counterparts, duplicated meiotic prophase chromosomes first appear as long thread-like structures in which the sister chromatids are so tightly glued together that they appear as one. So it is during early prophase 1 that the homologues begin to associate along their length in a process called pairing, which in some organisms at least begins with interaction between complementary DNA sequence, also pairing sites in the two homologues. So when the prophase progresses, the homologues become more closely juxtaposed, forming a four chromatid structures, also called bivalent. In most species, homologue pairs are then locked together by homologous recombination. DNA double-strand breaks are formed at several locations in each sister chromatid, resulting in large number of DNA recombination events between the homologues. So this is the concept behind it. Also, as we have already covered, I have just written down to make you understand, understand the, whenever you will go through the slide, you will be able to easily understand. So what happens when this homologous pairing will happen, there is some protein complex which will help in this pairing. This is called synaptomial complex. So the paired homologues are brought into close at a very close position or the juxtaposition with the structural axis or the axial cores about 400 nanometer apart by a mechanism that depends in most species on the double strand DNA breaks that occur in sister chromatids. So now the question is, what pulls this axis together? One possibility is that the large protein machine called a recombination complex, which assembles on a double strand break in a chromatid, binds the matching DNA sequence to the nearby homologue and helps reel in this partner. This so-called presynaptic alignment of the homologues is followed by synapses, in which the axial core of the homologue becomes tightly linked to the axial core of its partner by a closely packed array of transverse filament to create the synaptomial complex, which bridges a gap, but now only 100 nanometer between the homologues. Although crossing over begins before the synaptomial complex assembles. The final step occurs while the DNA is held in the complex. The morphological change that occur during homolog pairing are the basis for dividing meiotic profiles into five sequential stages: liptotin, zygotin, pachytin, diplotin, and diakinesis. So, profile starts with liptotin when homologs condense and pair and genetic recombination begins. At zygotin, what happens? Now, the synaptomial complex begins to assemble at sites where the homologs are closely associated. So the packaging stage can persist for days or longer until the desynapsis begins at diplotin with the disassembly of synaptomial complex and the concomitant condensation and shortening of the chromosome. Now, so whatever we were discussing, this is the pictorial view. So these are two homologous chromosomes. This is replicated paternal chromosome, and this is replicated maternal chromosome. This is centromeres. These are sister chromatids. This is bivalent. Yeah, so now when crossing over happens, they form chiasma. Homolog pairing and crossing over. So the structure formed by two closely aligned duplicated homologs are called is called bivalent. So as in mitosis, the sister chromatids in each homologs are tightly connected along their entire length as well as their centromeres. At this stage, the homologs are usually joined by a protein complex called synaptomial complex. A later stage, bivalent, in which a single crossover has occurred between the non-sister chromatids. It's only when the synaptomial complex disassemble and the paired homologs separate a little at the end of prophase 1 as shown, but the crossover is seen microscopically at the very thin connection between the homologs, also called chiasma. So this is a simplified schematic drawing of synaptomial complex. Here you can see multiple proteins are associated with this linkage between two homologous chromosomes. So this is one chromatid and this is another chromatid. This is chromatin loops or sister chromatids of one homolog. Here you can see axial cores of the two homologs. The protein cohesin proteins are cohesin proteins are there for condensation, and this distance is 100 nanometer. So there is some other proteins also, transverse filament and all, those are helping in this linkage. So we'll know it in a detailed picture. So this is the meiotic profiles where synaptomial complex formation is happening in budding yeast. So here also you can see the same picture like before. This is one chromatin loops of one chromatid. This is another chromatin loops of chromatids. Multiple proteins are there which are basically helping to make them linked to each other. So some elements are there. One is lateral elements, another one is transverse element, another one is central element. So lateral elements is consist of mainly HOF1 and RAID1 protein. Central elements is consist of GNC2 proteins, and transverse elements are consisting of ZIP1 protein. So these are all for yeast, but you have to understand that these proteins are very much evolutionarily conserved in between multiple organisms. So different homologs are present from yeast to mammals, and the functions are also similar. So again the same thing, but in a different stages manner. So here you can see homolog synapses and desynapses during the different stages of prophase. Here, so a single bivalent shown schematically at liptotin. The two sister chromatids collige and their chromatic loops extend out from a common axial core. Assembly of the synaptomial complex begins in early zygotin and is complete in pachytin. You can see the complex disassembles in diplotin. In the next slide, we are seeing that a bivalent with three chiasmata. So here you can see that the three sides of crossing over. So as I was mentioning that the number of crossing over can vary. It can be 1, 2, 3 or more, but these are very much regulated process. So different mechanisms are there to regulate the number of crossing over formation. So here this is a light micrograph of bivalent stage of grasshopper, and this is a schematic diagram of this thing. So you can see there are three chiasmata formation happen. So comparison of chromosome behavior in meiosis 1, meiosis 2 and mitosis. Chromosomes behave similarly in mitosis and meiosis 2, but they behave very differently in meiosis 1. In meiosis 1, the two sister kinetochores are located side by side on each homologue and attached to microtubules from the same spindle pole. The proteolytic cleavage of cohesin along the sister chromatid arms unglues the arms and resolves the crossover, allowing the duplicated homologues to separate at anaphase 1, while the residual cohesion at the centromeres keeps the sisters together. Cleavage of centromeric cohesion allow the sister chromatids to separate at anaphase 2. In mitosis, by contrast, the two sister kinetochores attach to microtubules from different spindle poles, and the two sister chromatids come apart at the start of anaphase and segregate into separate daughter nuclei. So this is how meiosis and mitosis differ. So in this case, you can see these two rates are homologous, two sister chromatids, and two grays are another two sister chromatids. But this red and gray are homologous chromosome. So you can see these kinetochores are attaching for one total chromosome or two sister chromatids. Kinetochores are attaching with them from one pole only. Here another kinetochore is getting attached to another homologue. So now these are blue are cohesin protein, which is helping them to link to each other. So in anaphase 1, this two will get separated to the different poles. And what happened now, you can see in metaphase 2, this now they will align in the equatorial position, and then this chromatids will get separated. So in this case, again the same rule of mitosis will be happen, that from one pole of one microtubule will getting attached to one with form a kinetochore with one sister chromatids, and then they will get dislocated to two different poles. So now the final result is that haploid daughter nuclei. Okay, so 1 2 1 2, and then they will be they will undergo telophase, and four distinct gametes or four distinct haploid daughter nuclei will be formed. But in mitosis, what we also saw in the previous class, that what is happening in mitosis, they don't try to find their homologous pairs, but they do different chromosomes will get aligned at the equatorial positions, and then what will happen, the microtubule will get attached with the sister chromatids, and then in the anaphase stage they will be dislocated into different poles. Now, as we were talking about homologous recombination, crossing over, now we have to understand how this homologous recombination, crossing over and all is happening. So the key feature of homologous recombination, which is also known as genetic recombination, is an exchange of DNA strand between a pair of homologous duplex DNA sequence. That is, segments of double helix that are very similar or identical in nucleotide sequence. So this exchange allows one stretch of duplex DNA to act as template to restore lost or damaged information on a second stage of duplex DNA. Because the template for repair is not limited to the strand complementary to that containing the damage, homologous recombination can repair many times the DNA damage. So also, as homologous recombination, we have seen that during DNA damage or DNA damage repair, homologous recombination is a way uh, to restore the DNA damage. So the same mechanism is applied in the meiosis also. So homologous recombination accurately corrects these accidents, and because they occur during nearly every round of DNA replication, this repair mechanism is essential for every proliferating cell. Homologous recombination is perhaps the most versatile DNA repair mechanisms available to the cell. The all-purpose nature of recombinator repair probably explains why its mechanism and the proteins that carry it out have been conserved in virtually all cells in Earth. So DNA base pairing, as we were discussing, guides this homologous recombination. So the hallmarks of homologous recombination is that it takes place only between DNA duplexes that have extensive ranges of sequence similarity or homology. Not surprisingly, base pairing underlies this requirement, and the two DNA duplexes that are undergoing homologous recombination sample each other's DNA sequence by engaging in extensive base pairing between a single strand of one DNA duplex and the complementary single strand of the other. The match need not to be perfect, but it must be very close for homologous recombination to succeed. So in its simple form, this type of base pairing interaction can be mimicked in a test tube by allowing DNA double helix to reform from its separated single strands. This process, also called DNA renaturation or hybridization, occurs when a rare random collision juxtapose a complementary nucleotide sequence on two matching DNA single strands. So as we have understood that homologous recombination is very crucial for meiosis. So it consists or it comprises of a group of reaction including broken end processing, strand exchange, so including broken end processing, strand exchange, limited DNA synthesis and ligation to exchange DNA sequences between two double helices of similar nucleotide sequence. Having discussed its role in accurately repairing damaged DNA, we now turn to homologous recombination as a means to generate DNA molecules that carry that carry novel combination of genes as a result of deliberate exchange of material between different chromosomes. So what happens now when this crossing over happens? So one strand gets exchanged with another strand of this homologous chromosome. So this occasionally occurs by accident in mitotic cells, but it's frequent and necessary in meiosis for recombination. Okay. So this homologous recombination occurs as an integral part of meiosis, whereby chromosomes are parceled out to germ cells. The parceled out means the chromosomes are now getting into from one cell we are getting four gametes. So now this is how the homologous recombination during meiosis produces chromosome crossing over and gene conversion, resulting in hybrid chromosomes that contain genetic information from both the maternal and paternal homologs. Crossing over and gene conversion are both generated by homologous recombination mechanisms that at their core resemble those used to repair double strand breaks. So we will understand this. So in meiotic recombination, what happens? It begins with a programmed double strand break. Okay, so homologous recombination in meiosis starts with a bold stroke. That is what happens. A specialized protein, also called SPO11 in budding yeast, breaks both strands of the DNA double helix. DNA double helix, you can see. So they break both the strands. Okay, both the strands in one of the recombining chromosomes, like SPO11 acts as a topoisomerase, and after catalyzing this reaction, this remains covalently bound to the broken DNA. So what happens? You can see the chromosome duplication and meiosis happened. Then You can see that after meiosis, four gametes or four nuclei has been formed. So here you can see these are the site of gene conversion, site of crossover. So in this one uh, homologous chromosome is painted as red and another is blue, so crossing over has been occurred, you can see. So they has formed this red blue patches. So in meiosis is the process by which a diploid cell gives rise to four haploid germ cells, and in which the paternal and also maternal information are present, and they are reassorted. Okay, they are reassorted to form some chromosome crossovers. So now coming to the mechanism, so these are two paired homologous chromosomes. PO11 MRE11 nucleus complex are helping in this recombination and double strand break. So what happens, this protein will bound to one chromosome and they cut and they start processing the end. So what is the processing in the end? After binding with it and after cutting it, they will then 5' to 3' endonuclease activity they will show. So they process, they cut through 5' to 3', and then they will start recombination. So Reiki like protein catalyzes the strand exchange, then they will do strand exchange. How it will be formed? It can be happen in two alternate ways. Okay, so what is happening when they are cutting this one strand, the other strand can, we know the homologous chromosomes are the bases are same, so they can form they can pair this one DNA strand can pair with the other DNA strand of another non-sister chromatid. Okay, so now they will, that will get replicated. So another two alternate pathways it can be done. One, when it will release from invading another strand, and it will then it will then take the other of its own DNA as a template and then replicate. And another thing can be happen, that it will capture the second strand. Okay, it will capture the second strand and it will go, and then the additional DNA synthesis happens, so then it will also replicates, and then they are forming a crossover. So additional DNA synthesis followed by DNA ligation. So here you can see that there is a crossover happened, and this is also called double holiday junction. So now the DNA strands will get ligated with DNA ligase. Also after crossing over, when it will be done, DNA strands cut, this DNA strand will be cut, and then two another DNAs will be formed with crossover. So you can see here the crossing over has happened. There is exchange of gene between red and yellow, but there is no crossing over happened, so there is no exchange of gene happened. So during meiosis, some abnormal chromosome dislocation or disjunction can be happened, and that may or may not lead to some physiological problem. So in normal condition in meiosis 1, what happens, two homologous chromosome will get separated, but when this normal condition is not there, there can be one thing, that in one cell only the both the homologous chromosome will go. Either achiasmatic non-disjunction can happen, that what will happen, that one sister chromatid and other sister chromatid or other chromosome will go together, which is not normal, or premature separation of chromatids, or it can happen that one there will be separation of chromatids which is premature. This is not supposed to be in the case of meiosis 1. In meiosis 2, the normal condition is that to separate two sister chromatids, but it can also be happen that they are not separating, so then the scenario is called non-disjunction. So this is all about meiosis, how it is differing from mitosis, how homologous recombination is important for meiosis, what is crossing over, what is chiasma, what is synaptomeal complex. Now we will briefly cover nuclear localization signals and how nuclear import and export happens. So we know that nuclear localization signals direct different nuclear proteins to nucleus. So what happens when proteins are experimentally extracted from nucleus and reintroduced into cytosol, even the very large ones reaccumulate efficiently in the nucleus. Sorting signals, also called nuclear localization signal, are responsible for the selectivity of this active nuclear import process. The signals have been precisely defined by using recombinant DNA technology for numerous nuclear proteins. In many nuclear proteins, the signals consist of one or two short sequences that are rich in positively charged amino acids, that is lysine and arginine, with a precise sequence varying for different proteins. Other nuclear proteins contain different signals, some of which are not yet characterized. So, nuclear localization signals can be located almost anywhere in the amino acid sequence and are thought to form loops or patches on the protein surface. Many function even when linked as short peptide to lysine side chains on the surface of a cytoplasmic protein, suggesting that the precise location of the signal within the amino acid sequence of a nuclear protein is not important. Moreover, as long as one of the protein subunits of a multi-component complex displays a nuclear localization signal, the entire complex will be imported into the nucleus. So it's important to have the nuclear localization signal, that is positively charged lysine and arginine. So this is how the nuclear pore complex looks. So this is, you can see two membranes of nuclear envelope. So this is a protein complex, this is membrane ring protein, this is a nuclear basket, disordered region of channel nucleoporins. So these are scaffold nucleoporins, this is channel nucleoporins. So this is how the nuclear pooling complex, or the NPC, looks. This is some cytosolic fibrils attached with the outer envelope of outer nuclear envelope. So the arrangement of nuclear pooling complex in the nuclear envelope, what happens, nuclear proteins are arranged with a striking 8-fold rotational symmetry. So in this case you can see a striking 8-fold rotational symmetry is there. And in addition, different microscopic studies showed that proteins that make up the central portion of the nucleopore complex are oriented symmetrically across a nuclear envelope, so that the nuclear and cytosolic sides looks identical, so that the nucleus and cytosolic sides look identical. Okay. The 8-fold rotational and 2-fold transverse symmetry explains how such huge structure can be formed only from 30 different proteins. So, based on the approximate localization of the central portion of the nuclear pore complex, nucleoproteins can be classified into transmembrane ring protein that spans the nuclear envelope and anchor to nuclear protein complex or nuclear pore complex to the envelope. Scaffold nucleoproteins that form layered ring structure. Some scaffold nucleoproteins are membrane bending proteins that stabilize the sharp membrane curvature where the nuclear envelope is penetrated. Channel nucleoproteins that line a central pore. In addition to folded domain that anchor the proteins in specific places, many channel nucleoproteins contain extensive unstructured regions where the polypeptide chains are intrinsically disordered. The central pore is filled with a tangled mesh of these disordered domains that blocks the passive diffusion of large macromolecules. So the disordered regions contain a large number of phenylalanine glycine repeats. Fibril protrude from both the cytosol and the nuclear sites of the NPC. By contrast to the two-fold transverse symmetry of the NPC core, the fibrils facing the cytosol and the nucleus are different. On the nuclear side, the fibrils converge at their distal end to form a basket-like structure. The precise arrangement of individual nucleoporins in the assembled NPC is still a matter of intense debate because atomic resolution analysis have been hindered by the sheer size and flexible nature of the NPC and by difficulties in purifying sufficient amount of homogeneous material. So basically this is how it looks. There is a 8-fold and 2-fold symmetry, and so these are the proteins associated with the nuclear pore complex, and there is also some cytosolic fibrils are attached with the outer nuclear membrane. So during nuclear import, so the different kinds of uh, nuclear import receptors are there which are binding to different cargo protein. So for import receptors, different nuclear import receptors bind to different nuclear localization signals and thereby different cargo proteins. Okay, so cargo proteins 4 require, here you can see cargo protein 4 requires an adapter protein also, which is green in color, to bind to its nuclear import receptor. So for cargo protein, the nuclear import receptor is this dark blue, and along with that it also requires nuclear import adapter protein. So they also contain a nuclear localization signal that binds them to an import receptor, but this signal only becomes exposed when they are loaded with a cargo protein. Now, as we were talking about the compartmentalization of RAN GDP and RAN GDP, so the localization of RAN GDP in the cytosol and RAN GDP in the nucleus results from the localizations of two RAN regulatory proteins, that is RAN GTPase activating protein, that is RAN GAP, RAN GTPase activating protein. What is GTPase? Who dephosphorylate GTP, okay, that is GTPase. So RAN GTPase activating protein, which is RAN GAP, which is located in cytosol, and the RAN guanine nucleotide exchange factor, that is RAN GEF, which transfers GDP to GDP, binds to chromatin and thereby located in the nucleus. So RAN GDP is imported into the nucleus by its own import receptor, which is specific for the GDP bound conformation of RAN. The RAN GDP receptor is structurally unrelated to the main family of nuclear transport receptor. However, it also binds to Hg receptors in NPC channel nucleopodes. So here you can see in RAN GEF what is happening. It is helping in getting bound of GDP with RAN and excluding GDP. And again, this RAN GDP, when it goes to cytosol, RAN GAP dephosphorylate the GDP and RAN GDP is forming. So now, how GDP hydrolysis by RAN in the cytosol provides directionality to nuclear import. So here you can see, for simplification, you can see this is a nuclear membrane, outer membrane, inner membrane. This is nuclear site, this is cytoplasmic sites. This complex structures are nuclear pores. So here the picture has shown nuclear import, and here the nuclear export. So what is happening, movement through the nuclear pore complex of loaded nuclear transport receptors occurs along the FG repeats displayed by certain NPC proteins. The differential localization of RAN GDP in the nucleus and RAN GDP in the cytosol provides directionality, these directions, okay, to both nuclear and export and import. So here as we can start, protein with nuclear localization signal. So this cargo we need to import to nucleus. So what will happen, it will be getting bound with nuclear import receptor, and also when it will get bound, then it will get bound with the cytosolic fibril, okay. So then when it will come inside it, RAN GDP will bound with it, and then the cargo will be delivered. So RAN GDP will export the nuclear import receptor outside. When it will be exported, what will happen, GDPase will act, and then it will form RAN GDP and phosphorus. So this is how the cycle goes. Also, in the case of export, what happens when the cargo is delivered, and but now the cargo has to be exported to nucleus, what happens, RAN GDP will bound with it, then RAN GDP will go to outside, and then the cargo will be delivered. When the GTPase will act and form RAN GDP plus phosphorus, then nucleus export receptor will be free, and then nuclear export receptor will again get recycled or gets delivered to the nucleus again. So this is how the nuclear localization acts. If you have any question, we can discuss. Or if you want to explain some of the points in another for another time, we will go through.
Hello, ma'am. Good evening.
Good evening. Yes.
Ma'am, in the ninth question of the first question answer series, you showed like
Mm-hmm.
for an experiment.
Mm-hmm.
Like, can you just explain briefly about that experiment? I have been seeing that experiment in some papers, and I don't. Even though I read about it, I didn't quite understand that. No, yeah, that one 7,
This.
slide number 7.
Oh, okay. This one.
Yeah,
Yeah, yeah,
yes.
sure, sure, sure. So, yes. So, what happens? You want to know, so you have a protein, okay, and you know that this protein goes into nucleus. This protein has some function which is associated with nucleus, so it has to be inside nucleus, like different transcription factors, different which helps in replication, so that all should be in between nucleus. So what they did basically, this is a SV40 virus T antigen which helps in uh, transcription activation or enhancement. So it should be inside the nucleus. So what they did, they found that there is a short peptidic sequence, that is lysine, 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 arginine, lysine, which helps in uh, localizing this protein inside the nucleus. So this is that time it was also discovered that this is a sequence which is nuclear localization signal. But they wanted to prove that if we disrupt any of the amino acid in the protein structure, what will happen? Will this protein get recognized with the nuclear importer, or will be recognized with the nuclear pore complex? So what they did, they mutated the sequence. So one lysine amino acid, they mutated to threonine, and then they did immunofluorescence. So for immunofluorescence, what we do, we checked with one antibody against the protein, and that antibody is attached with any fluorophore, so we can image it, okay. So when, see what they did, basically they transfect one cell, they transfected with a normal protein, that is wild type protein. And then what they did, they injected some antibodies which are against that protein. So antibody will go and bound with the protein, and those antibodies are also fluorophore tagged. So when they did fluorescence microscopy, they will be able to see the localization of their desired protein, because the antibody against these proteins are fluorophore tagged, they will give signal. So when we are getting signal from the antibody, that means the proteins are there. So then they did immunofluorescence, and they get to see. And how you will know that they are inside nucleus? Then they will stain the nucleus with DAPI. So if we see that the localization of DAPI and the our desired protein location same or nearby, we can see that these are in nucleus. So in the case of wild type expression, they could see the nuclear localization, but in the case of mutated protein, they could see that those proteins could not localize inside nucleus, but they are in cytosol, okay. So that is how they do immunofluorescence microscopy and see the localization of the protein is it's getting hampered or not. I hope it's clear now.
So even though it's a mutated sequence, that there is the presence of the mutated sequence in the cytosol, right? Like,
Yeah, yeah, so the sequence is mutated, no, so that's
yeah.
why it's not getting imported. So basically when the protein is getting imported, that domain should be recognized by the nuclear importer, okay. So that's why it's now no, it's now not able to go inside the uh, nucleus, but residing in the cytosol.
Okay, okay.
question. Uh, yes, Srimati. Any question, anyone? I think Srimati has raised her hand.
Good evening, ma'am. I have one question.
Yes.
In mitosis, you have shown that while splitting, that means when the chromosome split, due to the separate means the pulling action of this spindle fiber, sometimes what happen, one particular chromatin gets separated and it moves to one side,
Mm-hmm.
which causes trisomy.
Mm-hmm. Mm-hmm. Mm-hmm.
So my question is that is it natural or it happens due to some kind of mutation?
Yeah, no, it's not natural. So what happens due to different factors? So yeah, yeah, obviously due to mutations or so whatever proteins are actually acting no during this cell division, so those are basically responsible for taking care of this dissociations of synaptomial complex. And also if any mutation happens or the protein is not able to active properly, so this kind of dislocations happen. So this can be of due to mutation of any particular gene. This can be of due to environmental factor like UV radiation, so that will lead to DNA breakage, and those protein won't be able to recognize that breakage or something happens. So yes, so these are not normal, but due to mutations and different environmental factors, uh, like UV radiation or something, so obviously mutations. Sorry, I lost my connection. Bikram, could you hear me? Whatever I was telling.
Uh, Sorry, ma'am. After a few seconds,
I
it
got
got
lost.
disconnected. So,
Okay, so the point is that I think my screen is not getting shared. Okay. So the point is that no, it's not normal. It's due to different mutations, due to different DNA damage, and different environmental factors, UV radiation, and all. So whatever. So what happens in this? Whenever the chromosome dislocations happens, these are very regulated processes. Okay, so different proteins act here. So these proteins also get needs to get translated, transcripted. The gene should be expressed, so if there is any mutation happens, so those proteins if gets dysregulated, that will get hampered, and that the our next process will get hampered. So in that cases, you have seen in Klinefelter syndromes like Down syndromes. this kind of abnormality happens, no? So this is not normal, but obviously due to any kind of mutations or dysregulations of other genes.
Hi, ma'am.
Yes.
Hello, ma'am.
Yes, tell me.
Good evening, ma'am.
Good evening.
Can you hear me?
Yes, I can hear you.
Hello, ma'am.
Hi. Am I audible?
Yeah, ma'am.
Yeah, tell me.
I wanted the clear answer for the assignment 5, ninth question, ma'am. Pick the correct statement regarding nuclear organization.
Ninth question. Yeah,
Yes, ma'am.
yeah. So, do you know what is heterochromatin euchromatin?
No, ma'am.
Okay. So I will explain you. So what happens in the nucleus? We know that we have chromatin. Okay, and chromatin sometimes some part the chromatins are very condensation also are leading by some proteins which are getting condensed, and some parts are not condensed. So you know when the gene should be replicated, the gene should be transcripted. So transcription will be hampered if there are too many proteins. So I'm telling you in a very layman language, if there is very much crowding or trafficking, the transcription will get hampered, okay, logically. So in this heterochromatin region, these are darkly stained because those are condensed very tightly. So if some part of the chromosomes are very condensed, so those will be transcriptionally active or inactive. What do you say? Will be inactive because it is very much condensed. But when, and also this are darkly, when we stain our nucleus, so it seems that these are very darkly stained, okay. So the fact is that whenever the one area which is also called heterochromatin, which is darkly stained, is very much condensed, those area will be transcriptionally inactive. But here it is written that it is active, so this is false answer. The second answer is also true, because euchromatin is lightly stained. This is true. Euchromatin means the chromatin are not that much condensed, so it is lightly stained, and those are transcriptionally active because there is less crowding.
Okay, thank you, ma'am.
Yeah, and acetylation actually helps in euchromatic formation, not deacetylation. Okay, so
Okay, ma'am.
yeah, thank you. Your exam dates are not announced yet, right? I think it will be announced soon. So if you don't have any other question, we will stop today's class, and we again meet on next Tuesday from 6 to 8. We'll be discussing on week 6 content, and also if you have any doubt of this from week 1 to week 5, you can let me know. We will discuss, and also we'll discuss on some assignments.
Sure, ma'am. Thank you for your class.
Thank you everyone for the presence. Bye, see you.
Okay, ma'am. Bye.
Bye.